Hello, everyone. Welcome to our first session of the year of the Signals Research Suite Quarterly Connect Community Series. We released Signals Research Suite last year as a complete unified SaaS software for scientific research, and this is our inaugural Signals Research Suite Quarterly Connect, where we dig into the capabilities of Signals Research Suite that we think you will find useful in your research. My name is Chris Stump, and I'm a Product Marketing Manager for Signals Research Suite at Revity Signals, and I will be your moderator for this session. We have a fantastic webinar for you today, but before I introduce today's speaker, I want to share why I think you'll find this webinar useful. So what will you learn today? We assume that you already use Signals Notebook or Signals Research Suite, or you're thinking about doing so in the future. This webinar series will focus on Make, Test, Decide R&D workflows in Signals Research Suite, the first fully integrated SaaS platform for new biological and chemical modalities research. As many of you may know, we released Signals Research Suite last year, and it brings together Signals Notebook, Signals Vitro Vivo, and Signals Inventa into an end-to-end cloud-based R&D workflow platform with Spotfire and ChemDraw natively integrated. So today, this webinar will focus on Make, Test, Decide, highlighting antibody drug conjugate drug discovery workflows. You'll see how Signals Research Suite facilitates ADC research. This webinar will also address the importance of integrating various types of data. We then hope to have some time for Q&A. So our presenter today is Zev Vizovsky, Senior Principal Marketing Manager for Biologics and Signals Research Suite. Zev has a lot of experience in the biotech industry and worked at several startup companies where he focused on supporting scientists to get their science done more efficiently. He has also worked at life science software solution providers where he consulted and designed software solutions that met customer scientific and technical needs. Zev holds a PhD in neuroscience from the University of California, Riverside, where he worked on taste detection from molecular mechanisms to behavior outcomes in fruit flies. And before turning over the session to start us off, turning over to Zev to start us off, I have one quick housekeeping reminder before we start. I know many people are joining, and so you may want to watch the webinar later, but this webinar is being recorded, and the recording will be shared with all attendees in the next couple of days. So without further ado, I turn the floor over to you, Zev.
Great. Hopefully everyone can hear me. I really appreciate the introduction, Chris, and I'm excited to share our presentation with you all today. So before we jump into antibody drug conjugates, I think it's good to just understand the landscape. And when we think about, various different modalities going from kind of the smaller molecules all the way up to cells. So as drugs get larger, the degree of complexity in general increases from building these to tracking them and understanding mechanisms of action. And furthermore, notation or communication about each of these modalities is also increased in challenge. And furthermore, adding these modalities together, which I like to term, or maybe I haven't termed, Franken molecules, such as oligonucleotides conjugated to peptides or antibody drug conjugates, as we'll explore further today, only increases the need for tools that are sophisticated enough to support effective communication towards increasing the speed and accuracy of developing these drugs. So to make software powerful, it needs to communicate and represent science with the end goals in mind. For example, if you are working with small molecules as a chemist, you need chemical structures and other tools to showcase how structures are made, such as ChemDraw, which Signals is fundamentally integrated with. As you get larger, working as a scientist with oligonucleotides, for example, you need consistent and easy ways to represent and build, such as Helm or other sequence-focused tools. And as you get to the protein level, the scientist could need things like AlphaFold or other 3D structural tools. And also, although today we are focusing on antibody drug conjugates, it is challenging to find software tools to connect and support the needs across these modalities, as antibody drug conjugates are generally made up of three main components. The antibody, as most people know, are going to generate and bind targets of interest. We have the linker, could be chemical or peptide based, and then the small molecule warhead or drug that's acting on the target. And as I mentioned this note, because we're already starting out with potentially 3+ ways of representing groups' work, possibly with many softwares. And this puts communication and information as a key requirement, which can be challenging to accomplish. And furthermore, this is an example of the biotech we'll talk through today. Working on antibody drug conjugates also introduces various different scientific specialties. So you might need a potentially 3 or more separate subject matter experts that are groups which will work together to make the final candidates for testing. So here's our example team. We have Alex, who's working in the antibody drug discovery team. We have Alex and Stacy, the assay teams where primary and secondary screens are done. Maybe there's some decisions made at that stage. Then Paul, who is the protein engineering team, and Charles from the small molecule team. So we'll keep this in mind as the challenge to work seamlessly together when using various different tools that are not connected or easily searchable, 3+ data work streams and data sets being produced. And some of these cases need to be done simultaneously, or they are highly dependent with go and no-go decision for gating for the organization. And then again, in this slide, the ability for groups within the organization to be efficient, failing fast, and creating successful candidates are key metrics for drug discovery. So there's a lot here, and this is very challenging to do. So let's now review the antibody drug conjugate research flow, and then we'll show you how this is accomplished today in the Signals research suite. So first, we will typically do an immunization, which will harvest cells and then generate hybridoma cell lines for testing in our primary screens. This will generally be a high throughput screen like an ELISA or some sort of quick binding to check that we've gotten the antibody of interest. Then sending to a secondary screening. In this case, we'll be using SPR or other binding assays. And then from here, this is that first go or no-go decision. Do we need to start another immunization protocol, or do we have the candidates to move them forward? Assuming these meet criteria, then the candidate antibodies will be selected for further optimization, potentially expression, binding, humanization for antibody drug conjugation. And then once these antibodies are purified, they will need to be combined with the payload, the linker, to generate the antibody drug conjugate, which will again be tested extensively. And then finally, an antibody drug conjugate candidate nominated. Now, typically this is much more complex, but now this process can take, so for example, this is probably a more complex and more complete version of this. But this can take many, many months, and is much more complicated shown here than it is in the previous slide. So we'll be using the previous slide to walk through our example to give you a sense of what we can do, but obviously, there's more complexity to it.
Okay, so please take the poll here. So do you have the proper software to handle antibody drug discovery? And so we'll leave the poll open for about a minute. So about 30 seconds. Okay, we're going to close the poll. Okay.
That's great.
All right, so that's good. You can. I was expecting maybe we would get some results. No. Okay. Perfect. Perfect. Okay. So I just want to give you a chance to respond to that, Zev.
Yeah. Hold on 1 second. I think
Okay.
I need to see them.
So we've got about 25% of the people saying that they are that they have the proper software for antibody drug discovery. We have
Nice. Okay.
31% saying no, and then 44% saying not sure.
Yeah, no, I mean that's typically what we see. That's great to hear. I mean, obviously hard to hear. You know, we want to support you, but as we'll move through our presentation today, hopefully we'll be able to address some of those I don't knows and potentially knows for maybe how signals might support that.
Yeah.
All right, moving on. So let's go through our quick outline today. Let me click back into the right software. Here we go. So we've already talked through the background as I just gone through. We'll talk about discovering antibodies of interest, engineering them, creating linkers and payloads of interest, and then conjugating and tracking all of the binding and linker development, you know, via using things like Helm. We'll integrate and combine 3 major components, as I mentioned, for testing, potentially like a cell killing or other assays of interest. And then we'll end with that sort of full team to decide which of these antibody drug conjugates to promote. But instead of making you wait to the most exciting part, which I think is the decide, I'm going to show you that first. So what we are really, where we want to go and what we really hope to do is to look at a dashboard and be able to look at all of the data in a cohesive manner. So what we're looking at here is an example of a Signals Inventa dashboard, and it's powered by Spotfire, in which each of these columns have various different data we want to compare. So for example, many different candidates tested. You can see on the left, we can see the various different small molecule payloads. In blue, I'll talk about a little later in the talk, the linkers, and then there's some antibody information and assay data all connected. So this dashboard is kind of the everything all together. But to make this really useful, the next dashboard is what we want to end with, and you'll see this at the end of the talk, but I want to show you this now. So this data was assembled over various different months, and each of the constituents of the antibody drug conjugate, as well as the data that was gathered, when testing those. And overall, when we focus on the green bar or the green trend lines, these are ranges that represent sort of what we would consider a good candidate or a safe candidate or an effective candidate that falls within, the multi-parameter assessment that we need to do. And this allows us to compare over so many different metrics and assay data which candidates move through. And at the end, I'm showing you this because our goal for Drug Pipeline is to share this destination of where this presentation will take us. So, whether you're a scientist or a director level or a VP, this is the kind of data set that you'll be looking at when promoting candidates to move into development. All right, let's start off. So it begins with Alice. Alice is the antibody scientist who is going to the vivarium and to stimulate immunogenicity in the animals. And she uses the Signals notebook to prepare the animal experiment. She records the protocol, including the objectives and the steps that need to go into it. And one of the great strengths, of course, is it provides an environment in which all of the inputs like media, animals, proteins can be tracked and identified, linked together in a single experimental record. And this also provides a place where she can start recording the observations, in this case, intermediate, titer of the antibodies, and it's integrated with Spotfire directly so that these observations can be loaded into Spotfire and visualized, as well, as an example of a plate layout or samples that are being used. So it's all now part of a single environment that supports all workflows that scientists would do when they're generating data. from the animals. So the benefit or the value for Alice is that because she is responsible for multiple experiments in parallel, she has a single environment in which each of her experiments, each of those studies can be more easily tracked, the data for each of them organized. And additionally, now that she is, maybe getting toward the end of her hybridomas or ready to share those, she has the ability to, create those materials, and it's paramount as her hybridoma cell lines will be needed for testing within the next stages. Okay, so next, Alex, who is the assay scientist, will take the clones that have been selected and run them through a battery of assays. Again, in Signals Notebook, he will assemble the media, materials, equipments, and protocols that are used within the notebook. And then he will use vitro vivo experiment, and here we'll be dragging in primary activity data, and it'll support his end-to-end process because all of the data is integrated and centralized to track and for the data processing. So he'll be doing a high throughput ELISA raw data comes in, and the experimental properties, plate map, experimental details, and all of the contexts will be together in one place. And next he'll use, oh, I'm getting a little bit ahead. So here's the plate maps and all of that context. So next he'll use the high throughput screen QA/QC app, which we'll see in just a moment. And this can be applied to facilitate and normalize the viewing of the controlled plate layout. The results you'll see here are normalized. So we'll see all of the normalization that he's doing. And then these will then be applied to a hit pick or hit selection. And simply where in this case, 4 colonies appear to hit the criteria of interest and can be directly selected from here and sent back into the Signals notebook for the next steps of processing or queuing up any of the tasks that are needed to be performed downstream, like secondary screening. And so the true value for Alex is the ability to capture this mass amount of data that needs to be done seamlessly, and he has to screen thousands of these colonies. He has also having the process whereby he can drag and drop the results in. When I say drag and drop, typically these might also be done programmatically, so that's possible. And having the experimental record keep track of everything. And then having more or less automatic workflow that guides him through a step-by-step process and finally to the task requesting for secondary screening. Okay, so next the team will run a binding affinity assay, such as the surface plasmon resonance experiment, and Stacy is an assay scientist who will take those antibodies to do SPR assay. So we have the capabilities within our platform to run the SPR, processing the raw results and the final data across different instruments. And so in this example, she'll be bringing in it from like a BioCore instrument, walking through the vitro vivo experiment, and again, annotating these experimental properties. So the raw data is viewed and it goes through the normal steps performed, which are zeroing, which you can see here, against the SPR curve. Everything will be baselined against cropping. Let's see, I think we're there. There we go. Where she can take the full SPR scan in the flow cell and focus on the subset of space that actually has the data, and referencing, which has each of the flow cells can be correctly referenced against its reference flow cell for intra-flow cell data visualization, and then normalizing this data against the blank subtracted. So that will be here to normalize correctly for an empty flow cell data to get the final result. And finally, this data will then be put into the kinetic modeling. So she'll be doing the one-to-one mass transfer calculation to do a curve fit and be able to model the data and extract the right parameters. She can modify the way the curve interpretation is being done, and finally getting the kinetic curve here where you'll be able to look at the Ka and Kd binding coefficients, which those values results are useful to the team. And at this point, you could do QA, QC to knock out points or change as of interest. So after this, she can then publish this data, or you would want to publish this data in our publishing app directly into the data repository later to be integrated with other results. And then this would be assessed for the other antibodies who would be chosen to move forward. So that's what we are seeing here. And the real value for Stacy and the benefit is that this integrated platform allows for standardization and normalization of her data to be managed. Excuse me. So there are there's a bunch of different SPR instruments, as you may be familiar with, and each one of these has a different user interface and a different user experience. So, but by using Signals, she and her team can have a common experience and platform to support all of her data normalizations, any methodologies and workloads. So regardless of where the data is coming from, her team only has to generally learn a single interface for the data processing. And she has confidence because whether it's her who's processing it or someone else on her team, it will be done in the same manner. Okay, so having generated the hybridomas and getting the antibodies, the team is now ready to do a deciding of which antibodies are most efficacious for the various different requirements. So using Signals Inventa powered by Spotfire, the team will assess this with an antibody dashboard. So here we have access to a, a global search where antibodies can be selected and we can see all the individual results and CDR regions for each antibody. So that's going to be shown very shortly. With including any of the various different properties. So you can see that here as we're scrolling over. And then the lead antibody can be pinned and to compare the remaining antibodies to look for patterns of mutational differences between the lead antibody and other antibodies of interest. And in this case, we're using a key characteristic of HEC cell titer. So selecting the lead antibody allows for easy comparisons. But easier maybe would be to see a multiple sequence alignment. So as we move over to the multiple sequence alignment app, the scientists can then also pin a reference antibody and we can compare the CDR regions, which you can see here in red, and visualize the mutations within these regions directly to compare mutations from the lead antibody, again, looking at either HEC cell titer or other assay results of interest. And finally, what would be even better would be to have the system, I'll wait for this to load, is to have the system have an unbiased fashion for the best antibody by CDR region. And this is where the antibody component analysis, the user can select those that show, the most improvement in an assay of interest, in this case, maybe binding, and select those sequence patterns that are, driving these results of interest. So again, looking at the CDR regions or various different areas of interest. The best antibodies are then selected and sent back into the Signals environment. So that's what we're doing here. And the nomination can move this forward toward the antibody drug conjugate assembly. This group is, this list will be made available here in the Signals analysis object. And that includes all of the results, and they can be reviewed while the team decides how to progress the next steps forward of the overall project. So identifying the antibody, so the value for the team really here is to identify the antibody substitutions and correlate these to binding titer results of various different assays of interest, and to pick the best antibody efficiencies to move forward, or the effective antibodies to move forward. We can identify, which antibodies to advance, and the Signals Research Suite helps these results by integrating them over large data sets and providing antibody relevant tools, making this process easy and as transparent as possible. All right, next poll question.
Next poll question. So how many teams do you communicate with for your scientific findings? So the answers are 1, 2, 3, or 4 or more. And last time, I think I may have not given you enough time, so we'll make sure we give you ample time this time. So please go ahead and vote. And also, while you're voting, if you have questions, you can enter those into the question box so that we can have a Q&A with Zev at the end of the session. So about 30 more seconds for the poll question. Okay, about 5 seconds. Okay, we can close the poll and let's look at the results. Okay, so it looks like most people are seeing that they're communicating with at least 2 groups of people. Some are, so that's 58% Zev, and then the next largest group is 4 or more, that at 33%, and then the third group is 3, communicating with 3.
Yeah.
So you want to comment on that?
Yeah, I mean, I appreciate everyone's participation, and I think this kind of speaks to sort of an earlier point that I was making. You know, we talk about whether you're within one group or another and having to communicate effectively. You know, there's various different webs basically of communication, and having, you know, 3 or 4 different tool sets that each team uses, plus the 3 or 4 different teams that they have to communicate with, basically just magnifies this communication challenge. So appreciate that, and definitely gives validation to this idea that trying to simplify, whether that's through software or through, you know, ways to organize teams, is really helpful to speed up and make our scientists' lives easier. So I appreciate that.
Yeah, I appreciate the input, everyone.
All right, so we will move forward. Having identified the lead antibodies, next we want to optimize expression or potentially humanization. So Paul, the protein engineer, will now take the antibody sequence and put them into an expression vector. There's various different capabilities in Signals Notebook, but importantly, Paul is able to do his cloning, planning directly here. So we have integrated tools with SnapGene, best-in-class molecular biology tools. What we're looking at here, though, is in the Signals Notebook. So it's very easy for a molecular biologist like Paul to plan out their cloning experiments and use the cloning capabilities and tools to look at sequences, etc. He'll then be able to, you know, look at the various different primer sets, the plasmids, and develop the whole cloning experiment plan that will allow him to easily create expression vectors and perform the production. So within the Signals Notebook page, we have the ability to track all of the constituents, original plasmids, primers, insert sequence, and so forth, and even the final gel, so that you know you've generated the right sequence of interest. And Paul can use the worksheets in Signals Notebook to track the production experiment, including what steps have been completed, the timestamps, and that they've been fully completed. And this really guides the whole process end to end, from cloning through expression through purification. So this again, you know, is maybe an example of a little more of a manual version of the cloning, but this also can be done in a high throughput manner. And so the value for Paul is that, you know, he is going to benefit because Signals makes it very easy for the molecular biologist to sit down, you know, really plan out the experiment and use the capabilities to track the progress and drive those experiments in a consistent, reproducible, and reliable fashion. And so before we get into the next section, generally we will show the small molecule generation or process, but we don't have time today to do that. We have another presentation that's already been presented or showcased, and then it showcases this, and the recording can be accessed within, we have a library of videos. And so what we would ask you to do is please reach out to us or your sales rep, and we will make sure that you have access to this if you are of interest. But with that, we're going to move over and talk about the chemistry team, or the small molecule team, which will be developing the linkers. So Charles is responsible for developing the linker, and the linker for this organization is their special sauce. It differentiates them from other antibody drug conjugate companies. He needs to have a very easy and consistent way to draw the polypeptide or small molecule intermediate size molecules. And to do that, he'll be creating a new synthesis experiment, which you're seeing here, And we've integrated this with now Helm Monomer Editor, which allows the support of natural and customer curated monomers to be assembled into various different polymer chains of interest, natural, unnatural. We have the Pistoia alliance or any custom libraries that you, the customer, would be wanting to use. And this is the best-in-class capability to make sure that it's easy and possible to diagram and create quickly these novel polymers of DNA and RNA. And so with point and click, you can generate these. You can see here that when I hover over, you can see the various different chemical modifications. And this will be within the full synthesis process or protocol. And the various different structures will show up, so we can have all of this, at the top, and actually run this experiment, all in one place. And I think that's what I wanted to mention there. So as this loads, so the value for Charles is that it's really all about being able to correctly represent the linker, because it is their special sauce, and in this way, it's consistent across teams, from his conjugation chemistry team, but also across the organization, as other different team members, as we're collaborating with many different teams, need to also understand what does it mean to them, and I may need to visualize this in a different way. Signals with Helm support now provides that capability, and to be able to draw and manage and really standardize these complex set of monomers is required to represent these and other modalities. Okay, so finally the team will get together and they'll do the final assembly. And within Signals, they can also draw the antibody, or in this case, it's a segment, as I was mentioning, of the antibody of interest, and the linker chemistries, as well as the linker to the payload. And we can get a really accurate representation of the final ADC that's going into testing. I think that is a huge feat, because it is very challenging to get all these three teams looking at the same thing and really understanding what we're looking at. The team will then assemble together the three main components, as I mentioned, antibody, linker, payload, and this capability will bring it all together. So all of the components with the lineage, so knowing that this linker is connected to this particular ADC with the particular payload and the antibody, and all of those results that are connected is a very powerful and unique capability of our company to do the final assembly without losing any context. And so the key benefit for these, for the team and everyone, is this full traceability to allow for that final assembly that spans from small molecule all the way to antibody and basically anything in between, which is, again, a unique capability of Signals to support. All right. Just taking a drink of water as we jump into our sort of final scene, and this will kind of bring it all together. So this brings us back to the beginning, where I showed you the end dashboards. Here, not only do we have the antibody data, but we have the small molecule linker data sets all connected, as I mentioned. So we have, again, in Signals Inventa, various different dashboards of interest. We have a place where we can search, we can select various aspects of interest we want to dive into the data, look at all the data together for any comparisons. Instead of just querying for one thing, I'm querying for the antibody group conjugate, as well as the related antibody, linker, and everything else that's associated. And once this search is performed, you can observe the dashboard that looks like this. It's coming up. There we go. Where I was mentioning, we have the green, which is the final antibody drug conjugate test results. In blue is the linker results to the antibody, like load efficiency. The tan are the results of the antibody itself, and then the pink, although I wasn't able to show you this, are the small molecule payload results. So think about this just for a moment. What this is doing, it's pulling all the results together from disparate constituents that were all part of a single construct and assembling them into a single dashboard, a dashboard that every time I open it will refresh itself and the data reassembled. And within this, we can hover over and pin one of these reference sequences to see how the CDRs are changing or change between those, and get kind of this snapshot-like view. So comparing the payload versus the antibody drug conjugate might be really interesting, looking at the correlation analysis between the original payload test as well as the compared to the antibody drug conjugate. We can look at various different aspects of that. But most importantly, as I previously mentioned, and now you've seen this before, this is the multivariate space where we really need to see a tool that looks at over multi-parameter optimizations. So this parallel line chart and the green range is considered the good result for each of these axes. And this is really intended to make it easy for a scientist to select the antibody drug conjugates and see how they are performing across all of these axes to contribute. And this way, hovering brushes over the different antibiotic conjugates, and you can see what one looks like and hitting to the closest mark for the various different parameters. And this one is the winner, ADC 4162. And so, I think taking what would be a really complicated multi-parameter optimization space and providing a visual reference space for this really helps make data-driven decisions faster and easier. So this brings us to closer to the end of our presentation, and I think what I really wanted to do here was to summarize and mention that using signals for this antibody drug conjugate process can be driven rapidly and thoroughly from end to end. And this is all happening within the Signals Research Suite, which is a SaaS-based market-leading, which has market-leading capabilities. And we are continuing to grow and improve and add to what Signals can offer today to make it as easy as possible for customers like you to benefit from them, all in one user interface. And so with that, I'll end the in product part of the presentation, but we have one more question and then we will open it up for everyone else to ask questions.
Okay, let's see here. So the poll question: what percentage of your experiments are outsourced? So you've got a choice of 0 to 20%, 21 to 50%, 51 to 80%, and 81 to 100%. So we'll leave this open for about a minute and a half, just give everybody a chance to respond. And Zeb, while they're voting, I just want to tell you that we have a lot of good questions coming in. So I think you're going to have a very rich Q&A coming up.
I'm glad. I wanted to give us some extra time because I figured a lot of people would be interested in asking questions. So this is great to hear.
Yes. So it looks like right now, as the results are coming in, there's a tie for 0 to 20 and 21 to 50. So about 15 more seconds. Okay, about 5 more seconds. Okay. So yeah, it looks like we tied on, as I said, 0 to 20% outsourcing and 21 to 50%. And then about 11% of the attendees are seeing 51 to 80% of their work being outsourced.
Yeah.
And Zeb, I wanted to just put a plug in real quick before I give you a chance to respond to these. So we have an add-on capability that we are going to talk about at the next Signals Research Suite Connect. So and it deals with just give you a little teaser. It deals with CRO collaborations, and it's an add-on for Signals Notebook and also for Signals Research Suite. So yeah, please come to the next Signals Research Suite Connect, and you'll get a chance to hear about that. So go ahead, Zev. Let's hear.
Yeah. No, I mean, I think this is, it's definitely interesting. I would, I think probably maybe earlier or either in the year or last year, these numbers might be shifted a little bit to the right. So what I mean is more outsourcing happening, and it's possible that through various different economic climates and things like that, things could change. But I think it brings up a good point, which is, in some cases we do need to outsource, and it is a challenge because you're already, as I mentioned, you have your normal work stream, which could be 3 different softwares working with 3 different teams, and now you have to work with an external partner, a team, let alone if they're already set up in your procurement system or whatnot. You have a relationship with them, so I think it's just helpful for me to really, feel that pressure and the challenge of getting everything you want to get done while keeping timelines together. And it definitely resonates with me as previous scientists, the challenge that we're hoping to solve. So, Chris, I don't know if there's anything else you wanted to mention there, but I'm excited to talk about that more in our next connect.
Yeah, I mean, I think these poll questions are great input for us, just to, because we're trying, just like you said, we're trying to develop the software to kind of help researchers in the industry, and this gives us an indicator that we're heading in the right direction with the solutions that we're producing. Okay, so I think, Zev, we're ready for Q&A then. Okay.
Let's do it.
All righty. So I think I'm just going to go with the order at which these came in. So that means that some of the questions that I'm going to ask up front will be maybe towards the middle to the middle of your presentation.
That's okay. Yeah, we'll make it work. Let's have it.
Sounds good. Okay, so first, I'll take the first easy question, and I answered it in the text for everybody, but I know that people join the webinar kind of maybe after the webinar's already started, and people ask, well, will there be recording be available? And so what we're going to do in the next day or two is send out an email with a link to the webinar. So if you want to share it with anybody, you can do so.
Yep.
Okay, so let's jump into the questions. So you were showing kind of some of the animation of going through the kind of the workflows, and one person asked, can views be set up depending on a user profile? Say that they have team B that does not need to see nor change the full results from team A, and on a summary, but they would need to have full access to their data, but they would need full access to their data. Does that make sense?
I think so. Yeah. Let me see if I can restate it. So, if I understand the question correctly, as a scientist using the Signals Research Suite system, I don't want to be distracted by all of these, workflows or protocols or things that I don't actually do, but I need access to the data of the upstream team, or provide access to the downstream team, so that they can see the full context of whatever it's, whether it's experiment, the results, the metadata, the context of what I was doing. So that's what I heard. If that's not correct, happy to try to reach out afterward. But the answer to that is yes. The short answer is yes. We are able to really create a user experience that is very specific to the party or group who is doing the work, and even though it's generally a multi-group team that flows through this pipeline, you generally will only have to interact or work with your sort of next screen or next samples that are moving through, and generally will not be distracted by, like, I have all of these menu of options that I would never choose, so I'm just distracted by having to choose through them. And one thing I'll add on to this is we have many different organizations, and I know this is a question for a little bit later as well, but we have many organizations who work externally and have third parties have access to their Signals environment. And so in that regard, that's one of the other examples of how we would absolutely have to keep them, from a privacy standpoint, only able to look at their maybe chemistry, for example, work within the product and not see outside of that. So we can do that both within your organization as well as if you're potentially outsourcing. So I'll leave that answer there.
Okay.
But happy to follow up if I didn't hit the question on the note.
Yeah, that sounds great, Zev. I was having a little trouble seeing the full question, but I've been able to open that up a little bit. So I think you answered it well. So the next question, does this tool set have a scripting language to drive the GUI for workflows?
Ooh, okay. So does this tool set, I think we're referring to Signals Research Suite.
That's right, yeah.
Have a scripting language to drive the GUI, the user interface?
Right.
That's a really good question. I'm not exactly sure the why behind that. So are you, I guess this is hard to ask because I know the person is muted, but if you're interested in sort of like, can we do some of these things programmatically via APIs, if that is where the question is coming from, because I don't want to do things manually or want to be able to push information in seamlessly so that our scientists don't have to manually do that. The short answer is yes. If you're interested in more complex questioning like, what's your secret sauce and how are you coding this? That answer I won't be able to give you, mainly because I don't know it, but I'd be happy to flesh that out a little more if that is sort of the angle you were hoping to go. If you were trying to create maybe a custom application or something and you needed some more technical information.
Yeah,
but I'll leave it
I'm
at that.
seeing
That's
some
how
comments.
I interpreted that.
I'm seeing some comments in here, Zev, from the person who asked the question, saying to write macros, and I think he agreed with you. Yes, APIs and macros.
Okay, perfect. Yeah. So yes, and I mean, one of the last thing I'll just add on to that, Chris, and thanks for that context for the person who's asking. We have a bunch of our customers who have created their own custom applications on top of our software. So I'm saying this as we can support you through that process, but if you yourself are a consultant or someone who has that expertise, work within an organization that requires that, we don't stop you. We support you through it as needed, but you can definitely go off on your own and figure that out, and we will share the correct tool set or information or knowledge set that you require to do so. So I'll end it there, but appreciate that question. It's a really good one.
Yeah, so you got a thank you on that response, Zev, from the person who asked the question. So I think you answered the question, and I know that people use. I think you might have mentioned people use various programming language like Python and things like to do certain things as well.
Yep.
So yeah. So I guess the next question then is let's see here. Oh, okay. So this is probably an easy question here. Do you have a trial version of your software, even if the functionality is limited?
Yeah. So let's see. The short answer I think is yes for the most of it. We have a couple different ways to get your hands on it. So some of those we have some pre-baked walkthrough that you can access through our website, and I can share those. And then further, we have a more in-depth access. So it kind of depends on if you want to like fully spend maybe a couple hours or something like that. We have what we call a hyper trial, which would allow you to more or less have, I don't want to say it's unlimited access, but it's like some sort of an access for a various different amount of time, I think 2 or so weeks, and we would be happy to talk to you about that. So if that's sort of the way you're interested in, we have that kind of flavor or menu item. And if you're more interested in just like, I just want to kind of see a little bit more, I want to kind of have the ability to click through a little bit, but maybe in a more sort of guardrailed way, those both are depending on how much time you have. But Chris, anything to add to that?
Yeah, sure, Zev. So yeah, you're right. So on our website, we have like these mini demos that you can kind of follow along and kind of see how the software works. But and that's mainly centered on Signals Notebook. You'll see a lot of that, and then If you want to try, Zeb mentioned like a trial. We have like a 15-day trial for Signals Notebook that you can go on the website and put in, kind of your contact information, and you should be able to get set up within a couple minutes. That'll give you access to Signals Notebook. And then if you're more interested in trying out like the full Signals Research Suite, then the best bet is to probably reach out to your sales rep in your your local sales rep, and they can connect you.
Yeah, great question. We are always working to improve this process because we do understand from a buyer's persona how much you kind of want to get your hands on things, and we're trying to do our best to balance that with really understanding the time you have, the investment you have, and making the experience a really joyful one, so you're not frustrated with like, well, I don't know how to use the platform, so I need help to get to use it. And then so there's a little bit of a balancing act there to make sure we have it close enough to what it would look like in your environment while not spending as much effort, or balancing that effort on your end to make sure it's a light enough touch so you're able to see it and not feel overwhelmed.
Mm-hmm.
Yeah.
Yeah. Okay. So another question here is, when you have modifications to the amino acids, can you import the sequence and then add on the modifications, or do the amino acids need to be selected manually one by one?
Oh, this is a good question. So let me just make sure I'm summarizing correctly. So can you input modified bases, modified amino acids, peptides, etc., from the get-go? I believe the answer is yes. And I don't believe it requires you to modify them manually each time. However, you have the ability to modify them manually if you need to. But my understanding is the why behind this question would be, I don't have time to do that. I already have a library and I want to bulk upload or kind of move that in one go, and then be able to track who's using what or search for who's using what within various different sequences, peptides or oligos or nucleotides or etc. So if that is the why behind it, the answer is yes, you do have a way to basically take a library of interest, a custom library that's your secret sauce or whatnot, and upload that into the system, either by drag and drop, I think, or programmatically, and then you would have access to that library within that custom library within Signals Notebook using the Helm editor for your scientists or yourself to use in creating your sequences of need. So hopefully that's where I would answer that question.
So Zeb, I don't know that I interpreted the question any differently than you did, but one interpretation could be that like in the Helm editor, you have like the standard amino acids that you can just put in alanine, glycine, valine,
right.
And you can use those. But if you had like a non-standard amino acid,
non-natural or whatnot,
a non-natural one, you could define that
yes.
and then call it up, kind of a custom library, right? Is
yes.
That, yeah.
I think the way I just really quickly, the way I'm thinking about it is like, sometimes you have it all planned out, right? You got your whole library and you just wanted to put it in and then people to use it as you'd like. And then in some cases, there might be a really interesting piece of experiment. So you need the ability or the functionality to modify on the quote unquote fly, right? So I think both of those approaches are possible. And I wasn't sure which of those approaches fit the questions kind of behind the scenes of like why you're asking that, but both of those approaches are possible. I just wanted to make sure I tried to address sort of the like, oh man, this is going to be painful to put all this information in. I got to start from scratch and I already had this beautiful library done. Why can't I just kind of drag and drop it?
Yes. Yeah.
So anyway, I'll leave it there.
Okay. Very good. So just as a time check, we got about 8 minutes. So I need to just make a couple announcements at the end. So that gives us about 6, I think.
Perfect.
So there's a few questions in here that we might have to follow up on. I know that I see a question in here. It says, about confidentiality and SaaS-based software, and I know like, we have separate tenants and all that kind of stuff, but there's subtleties in all that, and so we probably should follow up on that question with the person
Yeah,
who
happy to
asked.
follow up.
Yep. Let's see here, and then let's see here. Okay, so can linker composition and identifiers come from an in-house small molecule database?
And linker composition and small molecule modifiers come from a custom database. I believe that's a kind of a similar question. Like if you have a particular data set or library of linkers or small molecules or various different modalities of interest that you want to bring into the system, the answer is yes, you should be able to bring those in. And I think without preaching the Pistoia Alliance and Helm, which I, by the way, agree with, being able to visualize those in a chemical manner or in a larger sequence, like a peptide-like string for the various different purposes is really the goal and the key. So, bringing those in, you can visualize them in the ChemDraw if is required, or modify that, etc. Or if you need to see that in a more sequence-like way, we can visualize that with Helm. Hopefully that answers your question.
Okay. Let's see here. The next so I guess the next question is that you didn't explain about linking to different data sources like storages on data warehouses or data lakes, or is there a way to connect programming language modules like R and Python? So there might be a couple questions in there, but maybe you want to address like the data sources first.
Excuse me. Sorry, I'm getting over a cold. You're right. I did not directly go into the technicalities of the data warehouses or data lakes. I would say the short answer is if we can connect it via API, it can be done. But the longer, more complex answer would be, fit for purpose, especially when people are doing, running their own like pipelines or statistical packages or things that they built with Python or R. I know they can be connected, so data can flow either out of or into your particular algorithm, I'll just say, for lack of a better term. But we would probably want to talk with you more carefully and figure out exactly I'm doing sequencing, or I'm doing, I need to run some, I don't know, like metabolomics or whatever those things are, which do require some more complex thought around how we want to bring that data in and out of the Signals platform. So I'll leave it at that. I know it's a little bit high level, but we'd love to talk to you more about that. So please reach out if we didn't answer that question. That's exactly you'd like.
Yeah, I think that's good, Zeb. I think we probably would have to maybe follow up on offline on that one, because there's some just to understand really what they're getting at.
Yep.
So the final question then here is it possible to add flow cytometry data to the notebook and research suite?
Oh my gosh, that's such a great question. We're actually talking right now and trying to figure out how to showcase this in another Connect. So the short answer is yes. And just to clarify the question, there are kind of two different aspects of flow cytometry, or FACS, right? One is the primary data capture, so that's not something that Signals Research Suite does right now. However, it is excellent and has great capabilities in the secondary screen, or the secondary analysis piece, which we would do through the Vitro Vivo or Inventa Spotfire powered. So yes, and that's great that you're asking that question, because that gives me some more excitement to talk about it in the future.
Good. That sounds like we got another topic.
Right?
Yes.
Wonderful.
Yes. All right. So I think we're running close to time here. So I want to thank all of you who have asked questions in here. You give us a lot of good discussion topics here. So as we close, I want to just mention a few announcements. So we plan to hold 4 of these Signals Research Suite Connect sessions throughout the year. We are also, just to give a plug for our Signals Notebook Connect and ChemDraw Connect, we have 6 of those each throughout the year. So you can go to our events and webinars section on our website to see the calendar kind of the events that we are holding, and also the in-person events that we are going to be at. So if you're in a particular area, say Boston, San Diego, or where there's conferences, maybe you could stop by and say hi to me or Zev. But for the Signals Research Suite Connect, today is our February 7th session. The next one's going to be June 26th, and I'll just give a plug for the CRO collaboration add-on capability that we're going to talk about. And then if you kind of liked what you saw and what you learned from Zev's presentation today, we also offer some training, kind of a little bit more in depth than kind of what you're seeing here. You're kind of seeing an overview of the capabilities, but if you need to learn how the ins and outs of the various aspects of the software, we have training for our Signals products. We have training for ChemDraw, and we have training for Spotfire. And so if you want to reach out to Stacy Fellows, her email there is on the screen. Also, you can reach out to your sales rep. They also are plugged into that. And then finally, we kind of hinted on that we have maybe a potential topic for the next session. But if you have ideas for topics for the next Signals Research Suite Connect, or even the ChemDraw Connect or the Signals Notebook Connect, just utilize my email address that you see there. Reach out and suggest some topics, and we'll try our best to kind of work those in. And so with that, I just want to thank you all again for joining us today for our very, very first Signals Research Suite Connect of the year. And also it's the very first Signals Research Suite Connect that we've ever had for 2024. And I want to thank Zev again for a very excellent presentation and great job with the Q&A today. And so, as I mentioned before, we'll share out the recording in the next couple of days. So watch your inboxes for it, and feel free to share the recording with your colleagues if you think this will be of value to them. And this concludes our session for today, and have a great day.